Okay, so today we're going to continue our discussion on cancer and genes involved in cancer, and we're going to focus particularly today on oncogenes. As you remember, there are two classes of genes that seem to be involved in cancer initiation and cancer progression. One are the oncogenes, which are involved in pushing cells to divide or inducing cell division, inducing uncontrolled cell division. And then we have the tumor suppressor genes, which are generally involved in inhibiting cell division. And what we find in many cancers is that the tumor suppressor genes are lost, they're inactivated, sometimes completely deleted from those cells, and so it allows the cells to continue to divide. I was looking at the number of slides I have prepared for today, and I think what I'm going to do is I'm only going to talk about the oncogenes. That'll take up most of the time. There will be a quiz at the end of this. And then next week, I will talk about, on Tuesday, I'll talk about tumor suppressor genes, what we know about them. There won't be a quiz. There won't be a quiz on either of the two lectures next week. And then on Thursday, I'll talk about some genetic technologies that are currently being used. So, in any case, today we're going to talk about the oncogenes. And we're going to talk a little bit about history, how they were identified, because I think it's an important piece of this, and it helps really understand how they function and what they do. So let's go ahead on to the next slide then. So the oncogenes were originally discovered associated with a class of viruses which are called retroviruses. There are many different kinds of retroviruses, but there's a subclass of retroviruses that cause cancer. Those are called RNA tumor viruses. In fact, the first RNA tumor virus that was discovered was back in 1911 by Peyton Rous. That was a retrovirus. And retroviruses have an unusual behavior. First of all, they have an RNA genome. And when retroviruses infect cells, they don't kill them. They carry out what's called a non-lytic infection. They don't kill the cells. This is very different than the flu virus or the coronaviruses like COVID-19, which when they infect cells, they kill a cell in the process of making more viruses. The RNA tumor viruses do not kill cells. In fact, if they killed the cells, they wouldn't be able to form tumors. So they infect the cells and they induce them to undergo cell division, but they don't kill them. So here's an outline of the infection. We'll go into a little bit more detail in a little bit. But basically, this shows a cell and the virus on the outside. The virus attaches to the surface of the cell, enters the cell. There's an RNA genome which is released, which I'll describe in the next slide. But more viruses are produced using the cellular machinery, the ribosomes of the cell, the protein synthesis of the cell, and more viruses are made, but rather than kill the cell, which is what the flu or the coronavirus does, they don't kill the cells, but they release the cells, they release the virus through budding. And that's what's shown, kind of try to show that in the drawing on the right-hand side. The cells survive, and the viruses are released through the membrane, and so the viruses actually have part of the cell membrane on the surface. So the point is, they undergo what's called a non-lytic infection. A lytic infection is where the cells bust open and the viruses are released. They undergo a non-lytic infection where they do not kill the cells, which is an important point because in order to get these cells to form tumors, they can't die, they have to survive. So let's look a little bit more detail at how these viruses behave. So if we look at these classes of retroviruses, these are RNA tumor viruses, so these are viruses that have an RNA genome and they form cancer. Again, the first one was described by Peyton Rous back in 1911. And as it turns out, there are two types of RNA tumor viruses. There's what's called a highly oncogenic retrovirus, and those cause cancer and basically kill the animal within 2 to 4 weeks. So they're very aggressive. This is the type of virus that Rous discovered and was later called Rous sarcoma virus. And then there are the weakly oncogenic retroviruses, which also cause cancer, but they cause cancer in a much longer time. It takes them around 6 to 9 months. So two types of RNA tumor viruses, highly oncogenic, 2 to 4 weeks, weakly oncogenic, 6 to 9 months, but they both cause cancer. So we look at the life cycle of the virus, because this is an important part of how they actually induce cancer. So let's look at what happens. So here on the left-hand side, I'm showing the virus, kind of that octagon shape. It's an RNA genome. The virus infects the cell, and then the virus is released into the cytoplasm. Sorry, not the virus. The RNA is released into the cytoplasm. The virus carries along with it a protein, which is called reverse transcriptase. And this is I've mentioned this before when we talked about the line elements, so you can see how these are in a way similar. The reverse transcriptase is part of the virus as it enters the cell, and once the RNA is released, the reverse transcriptase copies that RNA into double-stranded DNA. So we've gone from RNA genome to double-stranded DNA. Then there is also a protein which is called an integrase, again similar to line elements, and that integrates this double-stranded DNA randomly into the genome. There's no particular target. It goes randomly wherever it just happens to sit. So it cuts the double-stranded DNA of the genome and it introduces this double-stranded DNA from the virus. Once it's integrated, it's referred to as a provirus, and it becomes a stable part of the genome. A new set of genes have now been introduced into the DNA. Now the provirus has everything it needs. It has a promoter with a Tata box like we talked about earlier, and it has a transcription start site that is indicated by that little arrow above the provirus. And so it undergoes transcription like any gene, and it generates RNA, which is shown below the provirus. Now there's 3 different pathways that this viral RNA can take. On the very left-hand side, it shows it can undergo translation. Just like any other RNA that's made, it can undergo translation and generate proteins. Some of these proteins can have an influence on cell behavior. Some of these proteins can be involved in making new viruses. And so that's what's shown in the middle. It says packaged. So the RNA is then packaged into viral proteins that makes new virus particles that are then can be released through the membrane of the cell. And then finally, some of these RNA molecules, because there's reverse transcriptase in the cell, can undergo reverse transcription, making double-stranded DNA again, and then be reintroduced into the DNA in another place. So in principle, the introduction of one provirus can lead to many proviruses introduced into different sites in the genome because it continually is transcribing, undergoing reverse transcription, and reintegration into the DNA. So you can find these proviruses in many locations in the DNA. Okay, so let's look a little bit more closely at the proviruses. So the retrovirus has infected the cell, has undergone reverse transcription and integrated into the DNA in order to make a provirus. And from that provirus, as we saw, there can be RNA transcribed, which can do a number of different things, translate into different proteins, and be reintegrated into the genome through reverse transcription and reintegration. But at the top, what I'm showing is the structure of a classic provirus, prototypic provirus in a sense. So let me describe what's here. The squiggly lines represent the genomic DNA, the DNA of the cell. Then you see the provirus introduced, and it's flanked by two rectangular boxes, which are referred to as LTR, which stands for long terminal repeat. This long terminal repeat is, depending on the virus, can be 600 to 800 nucleotides in length, and they're identical on either side. So you see one LTR on the left and one LTR on the right. Within the LTR are all of the DNA sequences that are required for transcription. There's the Tata box, and then there is other sequences, DNA sequences, that allow various proteins to bind, which would activate transcription. And then the arrow represents the transcription start site. So transcription will go and then copy through make RNA through this provirus. Now notice that there's also one on the right-hand side. So there's a promoter and a start site. But that start site is going into genomic DNA. And that's going to be important a little bit later, because this virus can activate neighboring genes. But what's important here is that there are three particular genes on the provirus which are important. It's called the GAG genes, POL gene, and M gene. So the GAG genes are proteins that are required to make the viral coding when it makes a new virus. It's called capsid proteins that makes the new virus particle. The POL is the gene that encodes the reverse transcriptase enzyme. And the envelope, it's called envelope genes. These are proteins that are introduced into the cell membrane, which when the virus buds, through the membrane has these new proteins associated with it. So that's a classic provirus. Now, the Ralph sarcoma virus, which was discovered again in 1911, was really the first oncogenic RNA tumor virus, retrovirus, that was characterized. And it began being characterized in the 1970s when people started developing technologies that would allow them to analyze things better, like sequencing, protein analysis. And so the Rous sarcoma virus is an example of a highly oncogenic retrovirus. We saw that when we talked about Rous, because he made these extracts and introduced them into newborn chickens, and found that in 2 to 4 weeks they had tumors that basically killed them. So if we look at the provirus, and it behaves in the same way, it infects cells, undergoes reverse transcription, integrates into the genome, makes a provirus, and this is the provirus, a drawing of the provirus of Rous sarcoma virus. So we see there's the two LTRs, there's the GAG gene, the polymerase gene, the envelope gene, but in addition, there's another gene. This gene is called SARC. This is the oncogene. This is the gene that causes the virus to cause cancer. It is an additional gene that's found on this retrovirus. If you delete that gene or you mutate that gene, the virus will no longer cause cancer. So this is a single gene that commandeers the cell and causes it to begin dividing to form a tumor. This was the first oncogene ever discovered, and it was named oncogene because it's a gene that causes oncogenesis. So Rous sarcoma is a highly oncogenic retrovirus because it carries a gene that causes cancer of the oncogene. Now, since that time, there's been over 80, maybe 80, 90 different highly oncogenic retroviruses that have been identified. There's been viruses that are identified that cause tumors in chickens, different kinds of tumors, in birds, in mice, in rats, in monkeys. As far as I'm aware, there's never been a highly oncogenic retrovirus that's been identified in people. Probably because if it infects an early embryo at early stage, it's probably lethal. So there's been no human highly oncogenic retroviruses that have been identified, but they've all been identified in experimental animals. And each one carries a different oncogene. And down at the bottom I have a list. There's a gene called SARC, called sarcomas. A lot of these genes have given names that kind of come from the type of tumor or either the person that identified them. So it's a little bit of, it's like an acronym. So SARC causes sarcomas. That was found first in the Rao sarcoma virus. There's RAS, another oncogene found on a highly oncogenic retrovirus which causes sarcomas in rats. There is a gene called MYC, which causes a myelocytomatosis, which is a bone-type cancer in birds. MYB is one that causes a disease called myeloblastosis, which is kind of like a leukemia in birds and also in mice. REL is an oncogene also associated with a highly oncogenic retrovirus. Causes what's called a reticuloendotheliosis, which is similar to a lymphoma in birds. And there's a gene called ABL, causes tumors in mice. Named after the person who discovered it, which was John Abelson. And then CIS is another one, which causes sarcomas in monkeys or simian sarcoma virus. So all of these genes, and there's many, there's 80 or more highly oncogenic retroviruses. All of them carry an oncogene which is responsible for inducing the cancer. So here's the unnerving thing about all this, is that if you look at normal cells, normal chicken cells, normal mouse cells, normal rat cells, normal monkey cells, normal human cells, all have a normal cellular counterpart to these oncogenes. So normal cells have a gene that looks very much like SARC. Normal cells have genes that are RAS. Normal cells have a gene called MYC. So all of these genes that the retroviruses have are present in normal cells. They're not called oncogenes, they're referred to as proto-oncogenes. Because as they exist in the cells, they don't cause cancer. Apparently they're doing something else very important. But the way it's thought happened is that they were captured by the highly oncogenic retroviruses because of the way these things replicate and introduce themselves into the genome. That over time they have picked up genes, modified them ever so slightly, in order to make the virus more effective at replication. So, for example, RAS, we'll see later, RAS is a normal gene in the cell, doing something important, having to do with cell metabolism or cell division. Maybe controlling cell division in some way, normally. Viruses infect the cells, and on some rare event, due to the fact that it integrates into the genome, it may have picked up the RAS gene, changed it, maybe mutated it, or altered its expression in some way, or the SARC gene. And that gave the virus a selective advantage to replicate because it would induce somehow the cells to divide to make more virus. So the virus looks like it's using those genes that allow it to replicate better. So these are the oncogenes derived from normal cellular counterparts, which are called proto-oncogenes. So the highly oncogenic retroviruses, of which there are many, induce cancer in animals in a few weeks, very aggressive. And that's because they carry with them a gene, an oncogene, which induces the cancer process, or induces oncogenesis. But there's another class, which were the weakly oncogenic retroviruses. These also cause cancer. But they do not, it takes them much longer to induce the cancer. It takes 6 to 9 months versus 2 to 4 weeks. So what's different about them? Well, if we look at them, first of all, there's no oncogene associated with it. They look like a classic retrovirus with no additional oncogene. They infect cells. They undergo viral production, which is called viremia. That is, they introduce themselves into the cells. They produce virus. The virus is released from the cell, and then they undergo multiple infections so that the amount of virus in the animal builds up to a very high level. That's called viremia. Lots of virus in the blood. So they're undergoing many infections of many cells. And they also undergo multiple reintegrations because, as you saw earlier, the RNA can be reverse transcribed and then reintroduced into the genome. So the viruses, over this 6 to 9 month period, are replicating and producing more virus, reintegrating into the genome, more cells are being infected, and eventually they cause cancer. So how do they do that? Well, we look at one example. This is an example called Maloney, which is named after the person who discovered it. But more importantly, it's murine, murine leukemia virus. This virus causes leukemia in mice. It takes 6 to 9 months for the leukemia to appear. Now, if we look at the leukemic cells, and we purify the DNA, and we analyze the provirus, and analyze its location, what we see is something very interesting. If you look below, what you can see is the LTR, the GAG gene, the POL gene, the envelope gene, another LTR, classic retrovirus. But if you look at leukemic cells, right next to the LTR, the right-hand LTR, is the MYC gene. We've heard about MYC before because MYC was a gene that was found on a highly oncogenic retrovirus that I talked about in the previous slide. So what happened here is that the retrovirus integrated into the cell genome where the MYC gene was, and then the LTR on the right-hand side, which has a promoter, activates transcription of the MYC gene. Now, in some cases, the virus doesn't cause leukemia, it causes lymphoma, which is a different kind of cancer, but it also takes 6 to 9 months in mice. So now, if we look at lymphoma cells and isolate the DNA and analyze the viral provirus and ask what's unusual about it, turns out that in the tumor cells, when we look at the right-hand LTR, it's integrated next to the MYB gene. And again, the MYB gene is a gene that was originally described on a highly oncogenic retrovirus. In this case, when it integrates next to the MYB gene, it doesn't cause leukemia, it causes lymphoma. So the activation of MYC causes leukemia, the activation of MYB causes lymphoma. Now, these are not targeted mutations. That is, the virus is not entering the cell and immediately integrating next to the MYC gene or next to the MYB gene. What happens is, what's thought to happen is because the cells, it takes 6 to 9 months, because the virus is going through so much viremia, producing so much virus in the animal, and reintegrating so many times, hundreds of millions of times, that is, it's really just a matter of chance. And so ultimately it will integrate next to a gene like MYC or like MYB that when activated will cause leukemia or cause lymphoma. So it's a random integration. So it's integrating during viremia or integration, it's integrating all over the genome, many different places. Sometimes it's probably killing the cells. Sometimes it has no effect. But if it hits the MYC gene or hits the MYB gene, then the activation of that expression starts the cancer process. Now, what's interesting about this whole thing is that they're the same genes. So MYC was originally found on a highly oncogenic retrovirus. MYB was found on a highly oncogenic retrovirus, and so the mechanisms are different, although they are targeting the same genes, as if there is a specific class of genes that, when mutated or activated or disturbed, cause cancer. Now this is just two examples, leukemia lymphoma by murine leukemia virus, but there are many cancers in animals that are due to infections by these weakly oncogenic retroviruses, and in every case, the integration targets a specific different gene. Could be MYC, could be MYB, could be REL, could be RAS, there's many of these. And there is some evidence that there may be one very rare cancer, I believe it's called hairy cell leukemia, one very rare human cancer that may be due to a weakly oncogenic retrovirus. But all of these that I've talked about so far have been found in animal models, mice, rats, monkeys, birds. So that's the story with the RNA tumor viruses, the retroviruses. There are two types, highly oncogenic, weakly oncogenic, associated with either carrying specific genes derived from the cell or integrating next to specific genes in the cell. And that's all fine and good, but people started asking in the late 70s or so, what about human cancers? Does this have anything to do at all with human cancers? These are mouse models, animal models, viral models, so we may have learned some things about some of these genes, but is it relevant to human cancer? So people started looking for genes associated with human tumors, and in 1981, there was a very classic experiment that was done looking at bladder cancer. This was done by an individual named Weinberg, who was at MIT. And what Weinberg did in his group, they isolated bladder cancer biopsies, purified DNA from those biopsies, and then analyzed what genes were responsible for creating this cancer by DNA sequencing, by introducing them into cells, different kinds of techniques that were used to ultimately identify one or more genes that were responsible. Now, as it turned out, they did identify a gene, and the gene they identified was the RAS gene. Mutations in the RAS gene were associated with bladder cancer. Again, the very same gene that was identified by Harvey sarcoma virus, highly oncogenic retrovirus, in rats. So it turns out that, as far as we can tell so far, the genes that were identified in these experimental animal systems using the retroviruses, well, it may be that the very same genes are perturbed in human tumors. So what we identified in the retroviruses seems to hold over into non-viral human cancers. So as time has gone on, more and more of these genes have been identified in different tumor types, and it turns out they're the same genes. The RAS gene, the SARC gene, the MYC gene, the MYB gene, the REL gene, all the genes that were identified on these highly oncogenic and weakly oncogenic retroviruses turn out to be the very same genes that are disturbed or activated in human cancers, the same ones. And so we have the term proto-oncogene, which is the homologue of the genes that were identified on the highly oncogenic retroviruses, or the analogous gene in the cell, which is normal. And as it turns out, in human cells, in human cancers, these proto-oncogenes, which would be a proto-oncogene MYC, or proto-oncogene RAS, or proto-oncogene SARC, are mutated or activated in human cancers. So the same genes, not by retroviral induction, but by mutations or activated in some other way in human cancers, the very same genes. And the activation, as we see in human cancers, of these genes, turns out to be basically by three different ways. One is mutations. That is, you can get mutations in the RAS gene. That's what happened in bladder cancer. You can get amplification of certain genes, where DNA replication, you get more copies of the gene being made in the cell. And so when you get more copies of the gene, you get more RNA, more protein. This is seen in many cancers, particularly with MYC and MYB. We see massive amplification of these genes in some tumors. And then also, they can be activated by translocation, where you have one part of a chromosome moving on to another part of the chromosome, and that can have two different effects, basically, which I've drawn by the two arrows. One can be altered expression. We see that in human lymphomas. And the other can be altered gene product, altered protein, and that can occur, we see that occurring in leukemia cells. Now, early on, people had noticed that translocations were common in certain cancers. I mentioned that when I mentioned that timeline last time. So we're going to look a little bit at translocation just to give you an idea of kind of what they look like. But they can alter expression, or they can alter the protein product, which then changes its activity. So let's take a look at some of those. Here's an example of a translocation that's seen in Burkitt's lymphoma. This is a translocation that involves chromosome 8 and chromosome 14, and we'll look at the drawings in a second. But this is something that's extremely common in Burkitt lymphoma. It's present in probably over 90% of all Burkitt's lymphoma cases that have been observed. Now the thing is, Burkitt's lymphoma is a B-cell tumor. That is, these are B cells which are growing out of control. B cells are part of the immune system. They're in the blood, white blood cells. And B cells are the cells that produce antibody upon an infection. Now, as I mentioned, the translocation is present in probably well over 90% of the cases. And when you see some event like this, let's say a translocation in a tumor or in a certain particular kind of disease, you can ask the question: is this a consequence of the disease, or is it a cause of the disease? And generally, when we see something that's so common, over 90%, we usually think of it as causal. That is, it's somehow contributing to the development of the cancer. So it's an 8 to 14 translocation, so we're going to look at that. So if you look down, look towards the top, I drew out the chromosomes. Chromosome 8 is the white chromosome. Chromosome 14 is a smaller chromosome, which is kind of colored in. And the translocation involves the ends of each chromosome. You can see on the right-hand side, where part of chromosome 14, it's called a reciprocal translocation. Part of chromosome 14 has moved on to chromosome 8, and part of chromosome 8 has moved on to chromosome 14. Now, when people saw that this translocation was so common, what they did was they isolated that region of the fusion, that is, on chromosome 8 and 14, where the new parts join. They isolated that region and sequenced it to try and identify if there was something unusual there, and sure enough, what was found were two known genes. One was the gene for IGH, which maps to chromosome 14, and the other was the gene for MYC, which was found in chromosome 8. Now, I want to point out again, this is the MYC gene, the very same gene that was identified by the highly oncogenic retroviruses a number of years previously. And so when people found that this translocation in Burkitt's lymphoma involved the MYC gene, it was actually a pretty exciting finding, and again makes us realize that there's not an unlimited or very enormous number of genes that are involved in cancer, but that there's a smaller number, and that the highly oncogenic retroviruses and the weakly oncogenic retroviruses were able to somehow identify which genes and target the genes that stimulated cancer. So what happens during this translocation is that part of chromosome 8 moves on to part of chromosome 14, and below, where have the line drawings that kind of illustrates what's going on here. So I have the word MYC written, so this represents the MYC gene on chromosome 8. And so the MYC gene is a rather small gene that only has 3 exons, exon 1, exon 2, and exon 3. And remember, the exons contain the coding sequence for the gene, for the protein, and eventually the introns will be removed and the exons will be joined together. So exon 1, 2, and 3. And the start site for protein synthesis, the first AUG, is located within exon 2. So what happens during the translocation is that the breakpoint in chromosome 8 splits the gene between exons 1 and 2. So you have part of intron 1, exon 2, exon 3 being brought over to chromosome 14. And the breakpoint in chromosome 14 is such that the new fusion gene, contains the promoter and start site for the IGH, and I'll describe that in a second, for the IGH gene, linked now to exons 2 and 3 of the MYC gene. So what's the implication here? Well, IGH encodes an antibody. There's a number of different types of antibodies that are encoded by the B cells. One of them is called immunoglobulin H. It's one particular type of antibody that's produced upon infection. And since B cells, what they do is they produce the antibody, the IGH promoter is a very strong promoter. So what's happened, and so what's happened in this situation is that exons 2 and 3 are now linked to a very strong promoter, which is IGH, and it drives the expression of this new fusion gene. So I've written there high level of expression. You end up getting high level of expression of this unusual RNA transcript. But since the AUG is within exon 2, the messenger RNA that's made is competent or able to translate the MYC protein. And so you get an enormously high amount of MYC protein, and the MYC protein drives cell division. So the end result of this translocation then is bringing the MYC gene under control of the IGH promoter, which is overexpressed, expressed at high levels in B cells, leading to a high, very elevated amount of MYC protein in the cell, and that is oncogenic and drives cell division. Now, it's not a coincidence, all these things. Why we get B cell tumors, because the translocation is occurring in a cell where you end up getting high amount of the MYC protein because it's linked to an IGH promoter, and in B cells, which produce antibody, leads to very high expression. If the same translocation were to occur, let's say, in a liver cell or a skin cell, we would not get that elevated expression of the MYC because those cells don't express antibody. So the antibody expression drives the expression of the MYC gene. Now, in a sense, it's very analogous. If you look back at the drawing of the Winkley oncogenic retroviruses, where you have overexpression of MYC, in a sense, it's analogous because the LTR from the retrovirus has integrated next to the MYC gene and caused it to be overexpressed, leading to the tumor, in the same way that this translocation is causing MYC to be overexpressed, leading to the tumor. So here we have an example of a translocation leading to overexpression of an oncogene. Let's look at a different type of translocation. Okay, so this next translocation is a little bit more complicated. We'll walk through it. But ultimately, this translocation is going to lead to a altered or modified protein. In the case of the MYC expression with the translocation, it didn't change the structure or the function of the protein. It only changed the amount of the protein in the cell. So this is a translocation that's seen, and again, over 90-95% of chronic myelogenous leukemia, CML. It's a 9 to 22 translocation where chromosome 9, there's a switch, chromosome 9 moves on to chromosome 22, and chromosome 22 moves on to 9. This is referred to as the Philadelphia chromosome. It was first discovered in a lab in Philadelphia, and it is basically a diagnostic for chronic myelogenous leukemia. This is how the disease is diagnosed. They do karyotypes and look, and if there's a translocation, they immediately call it CML. So here we have two chromosomes shown below. Again, chromosome 9 is the white one. Chromosome 22 is darkened, a little bit smaller. You see the translocation there on the right-hand side, where part of chromosome 22 moves on to 9, part of 9 moves on to chromosome 22, and again, the chromosome breakpoint involves a gene that's already been identified, ABL. Yep. Again, we go back to the highly oncogenic retroviruses. Same genes involved in those cancers are now found to be involved in translocations in human cancers. So here we have the ABL gene being fused to another region, which is called BCR. BCR just stands for breakpoint region. So we have this translocation, and I'll show you below in the middle. Again, it is more complicated, so we'll go through it a little bit slowly. First, you have the ABL gene, which is on chromosome 9. There are 11 exons. And then we look at the BCR gene, which has 12 exons. And uniformly, in all of these translocations, the breakpoint occurs between exon 6 and exon 7. So when you get the translocation, the movement of chromosome 9 fusing with chromosome 22, what happens is the break occurs upstream of exon 1 and ABL and brings it over just downstream of exon 6. So you end up getting this fusion gene, which is shown below, where you have the BCR gene. The exons are shown darkened. You have 1, 2, 3, 4, 5, 6, and then it continues on, starting with exon 1 of the ABL gene, so you get this really longer transcript, and that actually ends up making a novel protein, which is called a fusion protein. So transcription starts at the arrow, makes the RNA, the splicing takes place, and the first part of the protein is the BCR protein, and then it continues on and translates from the white exons from the ABL gene and makes a much larger protein. It's called a BCR-ABL fusion protein. And that fusion protein has a novel function, and it forces the cells to divide. It's a trigger for cell division. Now, because this is a novel fusion protein, which is only seen in chronic myelogenous leukemia, to this translocation, people thought, well, this is a protein that's only present in tumor cells. It's not present in normal cells. So maybe we can find a drug or a molecule or a compound that would specifically interact with this fusion protein and block its activity. And sure enough, this was the first example of the identification of a drug which specifically targeted a cancer-specific protein, and the drug is called Gleevec. You can look that up. It's spelled Gleevec. Gleevec is a targeted drug that specifically attacks the BCR-ABL fusion protein and is a very powerful drug for treating CML. Highly specific. So you can see how once we know what the specific protein is, that we can start to maybe identify specific treatments for these cancers. Okay, move to the next slide. So what are the functions of these genes, these oncogenes that when deregulated or modified, push the cells to divide? And what are the function of the proto-oncogenes? The normal cells, the normal genes that exist in cells. What is their function? And how do they contribute to cell physiology? So what we've learned is that the proto-oncogenes function to signal cells to divide. That they're involved in all these signaling pathways that ultimately induce the cell to undergo cell division. So let's say we have, and if you look at this drawing, we'll kind of try and explain that. Let's say you have a cell that's in G0 that's not dividing. And we get a signaling molecule like a hormone that when the cells are treated with this hormone or signaling molecule, it could be growth factor or protein, these molecules bind to receptors on the cell surface. And that's what I've shown here. So there's the outer portion of the cell membrane, and we have the inner portion, which is the nucleus, and I just have one line drawn. It says genes, and that Y-shaped structure on the cell surface represents a receptor, which would bind to a specific signaling molecule like a growth factor or a hormone. So the signaling molecule will bind to the receptor, and what happens is the receptor sends a signal into the cell. And these signals are ultimately transported into the nucleus in order to turn on genes that are required for cell division. And what people have found is that the signaling pathway involves the interaction of a number of different proteins that eventually make their way into the nucleus. So we may have protein A, protein B, protein C, protein D, which are proteins which interact with each other, and a signal is passed from one to the next, from A to B, B to C, C to D, and eventually gets gets into the nucleus. A protein or signal will then bind to a promoter and turn on genes which are involved in cell division. Now, the function of these proto-oncogenes was actually came from two different lines of research. At about the same time that people were interested in understanding what these genes do, there was a whole other field of research, which is still quite active, and that is trying to understand the molecules, the proteins, that are involved in signaling a normal cell. People who were studying cell division and the regulation. And as it turns out, many of these proteins that are involved in this signaling pathway are the very same proteins as the oncogenes. So let me just show you that in the next slide. So in this slide, it's basically the same thing. What I've showed you here is rather than saying a signaling molecule A, B, C, D, and E, I've replaced those with the names of the genes that were identified to be part of that signaling pathway. And as it turns out, they are the same genes as the proto-oncogenes. So, in this one example, and there's many examples, the platelet-derived growth factor, which is a growth factor which stimulates cell division. If you sequence that gene and you sequence the cis oncogene, the same. If you look at the epidermal growth factor receptor, which is a receptor which is required to induce cell division, you sequence that gene and you sequence the ERB gene, another oncogene identified by the highly oncogenic retroviruses, same gene. The MET gene, the SARC gene, the first gene ever discovered from the Route sarcoma virus. It turns out sequence genes that are part of this pathway, unknown genes. Well, you know they're part of the pathway, and compared to sequences in the database, SARC comes up. The RAS gene, a signaling gene, the APL gene, a signaling gene. So all these signaling proteins that are involved in normal cell growth turn out to be the very same genes as the proto-oncogenes. Now, if you look in the nucleus, there's a few there that are transcription factors. That is, they bind to DNA and regulate transcription. So MYC is one, REL is one, MYB is another one. These are, the proto-oncogenes are normal genes. If you look at a cell, normal cell and you induce it to undergo cell division, and you look at the level of expression of the MYC gene, you see that it goes up, binds to promoters of genes which are required to complete the cell cycle. So this explains, in a sense, if we look at MYC, and all the examples I gave you, with the weakly oncogenic retroviruses, it was the overexpression of MYC by the introduction of the LTR. In a number of cancers, which I mentioned, leukemias and lymphomas, there is an amplification of the MYC gene. It means it's overexpressed. And the same is true with the translocation in the Burkitt lymphoma. There's a translocation that causes the MYC gene to be overexpressed. So you don't need to have mutations in MYC or any kind of a modification of the protein. All that needs to happen is to get elevated levels of the MYC protein, and then the cell basically thinks it's supposed to divide because the MYC gene goes up when cells are stimulated to divide, binds to the genes that it normally binds to, and then allows the cells to divide. The same is true with REL, the same is true with MYB. It's a little bit different when we look at the other genes in the pathway, the upstream genes. If you look at RAS, for example, RAS is an important carrier, a signaling molecule involved in normal cell division, and in the cancer cells, there's a mutation. And the mutation causes that RAS protein to be constitutively active, meaning active all the time, and passing the signal onto the next protein in the pathway, even though the normal signal is not present, but the RAS gene is acting as if. So that's what these oncogenes are doing, the proto-oncogenes are doing. They're normally involved in cell division, and when they're mutated, overexpressed, they contribute to the inappropriate cell division in cancer. Now, where we're going with all this is that now that we know what these genes are, we know what these proteins do, we can model them, computer modeling, look at their structure, know where the mutations are, and we can try and identify, or pharmaceutical companies can try and identify compounds that can specifically target these defective proteins. So if you have a mutation in RAS, can there be a specific compound that would interact with that protein? And that's what was done with Gleevec. The first one that was identified, that was, it interacts specifically with that BCR-ABL fusion protein. Another one that came out is called Herceptin, which binds to ERB, or the epidermal growth factor receptor, which is defective in many breast cancers. Tamoxifen is another one, which binds to the estrogen receptor. We talked about that earlier. We talked about gene expression. And there's a whole series of other ones. I just give you another example. This one, PF23-41066, is a drug that interacts with the mutated MET gene. So these are targeted drugs for targeted anti-cancer therapy. So we are going to stop here. That's what I want to say about oncogenes. Hopefully you have a better idea of what these all mean. There is a quiz after this, don't forget that. And then next time we will start talking about the tumor suppressor genes. That'll be next Tuesday.